Elliott is chairman of the Department of Biology at Caltech. And I'm not going to read his list of honors and achievements. It's in the booklet, and it's going to take way too long. Instead, I'll just briefly say that I think we can measure someone's contributions to science in some ways by imagining what the field would be like if they had never come along. And so thinking about what the telomere field would be like without Liz Blackburn, for me, my PhD advisor, is much like imagining what plant biology would be like without Elliott. It's impossible to imagine what it would be like. So in the early 80s, I'm sure many of you know that there was a lot of question about whether it was really useful to have a model organism to study plant biology. And really a lot of controversy about whether this was even a good idea, which just seems unthinkable now that Elliott's sort of shown us through Arabidopsis research what we can learn about some incredibly disparate fields, floral development, hormone signaling, even epigenetics. So I'll turn this over to Elliott and again say thank you so much for coming, Elliott. Looking forward very much to hearing both of your presentations today and tomorrow. Thanks.
Well, thank you very much. It's a great pleasure. Can you hear me if I walk away from the microphone? Back, okay, good. I've learned to bellow in 30 years of teaching. What my colleague Seymour Benzer used to call the casting of false pearls before real swine. I'm being taped. I better cut this stick. So here's the title of the seminar. They said, "What's the title?" I said, "All right, here's what I'll talk about. I want to talk about two things: the science of what we're trying to do to figure out how it is that plants grow in a particular way, that is, from their shoot apical meristems, that I'll introduce in a minute. But at the same time, I wanted to give some sense that the field is changing and that there's a way to do this that up until now has been quite successful, but there's a new set of technologies that captures the time element and certain levels of spatial resolution in analyzing the plants better than we used to be able to, and so that there's a new combination of techniques that gives us another leg up in trying to understand this difficult problem of how organisms work, a basic biological problem. And we've been, in my lab, calling this new approach computational morphodynamics, clearly because of a deficit of imagination. And so I want to, in part, talk about how plants grow and talk a little bit about some older work that we've done to understand how this collection of stem cells, the shoot apical meristem, gives rise to the above ground parts of plants, but at the same time I want to give a sense of how changing the methodology and including live imaging and computational modeling has enabled us to do some things that we couldn't have imagined doing before and has given us a new level of understanding of some of the processes that are going on during plant growth. Now, of course, this is the sober scientist version of a title, but as I was working on this seminar over the course of the week, I realized the title I had sent was a little bit sober, given my brief here, which is to give a general talk about this particular type of science. And so I came up with a second title, Too Late to be put in the pamphlet that comes here for this particular, for the Store Life Sciences lectures. And so this is the real title of this seminar. And so I want to begin with a few slides telling you why I say this. I mean, you probably thought it was love or money or something like that. It's the shoot apical meristem. And this is what everybody should be worried about and what we should all be concerned with in our laboratories. And so what's the shoot apical meristem? This is a Arabidopsis plant, an old picture I took many years ago of the plant that I'll be talking about. And it's not different from any other plant, except it's smaller, and so it's easy to use in a laboratory, and that's really why we use it. And it's a member of the mustard family, and when it begins to grow, it germinates from a seed. Can you see that in back? That one's a little more steady. Germinates from a seed and starts to grow, and after the seed grows, there's a couple of cotyledons. It's a dicot. And then the plant starts to make a series of leaves in this rosette pattern. And then when it's time to flower, the stem starts elongating, and there's still a few leaves that get made. And then there's a group of flowers made at the top. And I want to concentrate your attention on a little collection of stem cells that sits right up there in the middle of where all these flowers are being made in that picture. Earlier in the life of the plant, it would sit in the middle of the rosette of leaves where it's producing the primordia for each of the individual leaves. And so it's as true in Arabidopsis as in a sequoia that the entire above ground part of the plant derives from this collection of stem cells, which is formed in the embryo, as I'll show you in a second, and sits there and over the life of the plant generates time after time, leaf, 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 generates stem behind it, generates the flowers later in the life of the plant, and everything that's above ground in the plant comes from this population of cells. In an Arabidopsis plant, we're in a bigger plant. It's not to say there's no cell division after the cells leave the shoot apical meristem. There's a lot. But the specification of what they're going to be is pretty much laid down right in this little tiny group of cells. Here's a picture of it in a scanning electron micrograph. You can see individual cells that are about 5 micrometers across. And this is the shoot apical meristem. It's a stem cell population right at the very tip of each shoot. And in the time when this picture was being taken, the plant was making flowers. And so you can see in the region peripheral to the shoot apical meristem, called the peripheral zone, there are new primordia being made of flowers, one after the other, so that this one was made earlier, and then this one, then this, then this, then this. So you can see what a very young flower primordium looks like. And at the same time, the cells in the shoot apical meristem are dividing and pouring out cells beneath them, so that the meristem gets carried higher, and the stem underneath it gets established by the cells that come out of the bottom of the meristem. So it's making the stem underneath it, and on its side it throws out first leaves and then it throws out flowers. So this is a L systems view using some software we got from Przinkowicz with some rules that I'll be talking about tomorrow's seminar, not today's, to give you the idea of how this thing works, which is that the cells, the ultimate stem cell population, is at the very tip, and it's about 20 cells at the very tip of the shoot apical meristem, and these cells divide inside and on the surface where we're seeing. And if you hold the top of the shoot apical meristem at a steady point, it looks like the rest of the plant is pouring out behind it like a frozen fountain. What's actually happening is the meristem is going up as it puts out cells beneath it, which continue to divide and which enlarge and make the stem and push the meristem ever higher, so that in that redwood can get pushed up 300 feet above the ground. And so it's a very dynamic structure, and I want you to keep this in mind because that's where the puzzle comes in. If you look at patterns of gene expression in the Singer leaves, you get these successive leaf formations at particular places. I'll show you some patterns of activation of genes in these cells, and what you'll see throughout is that if you label with some gene like clavata1, and you can see the RNA for that is made in the core of the meristem, and the cells pour through that gene expression domain. It isn't constant, but the cells move through it. They come from the top where that gene is off. They get a little lower, the gene goes on. They get a little lower yet, and the gene goes off again. And so you have a constant pattern of gene expression with a flowing and changing pattern of cells going through this constant pattern of gene activation, which is a very hard thing to understand. And it's what I'm going to be talking about and why we need computational morphogenesis to help us solve this problem. So I want to say why the shoot apical meristem is the most important thing in the world. It really is, you know. It's first of all, from just a basic science point of view, it's the ultimate green machine, and it's a nanomachine. It consists of a few hundred cells, each about 5 micrometers across. Each cell has something like 100,000 to 1 million moving parts, more parts than a 777 aircraft, and those aren't moving parts. In the best of situations. Most of them shouldn't be moving relative to each other. These ones are all moving to each other, relative to each other. That is, the parts being proteins and other macromolecules and the macromolecular machines they make. So each one of these cells is more complicated than the most complicated objects that human beings know how to make, like this space shuttle. And yet it's only 5 micrometers across instead of being hundreds of feet long. And then what the meristem does with this enormous complexity of a few hundred cells, each with a few 100 million moving parts in this little thing, that it pours out behind it all the above ground parts of the plant, whatever it is, leaves, the stems, the flowers, the fruit and grain, the wood, the fiber, anything that we get from plants comes from a shoot apical meristem, except for roots, which come from a root apical meristem, which is organized rather differently. That's the second most important thing in the world. If you're into money, and since this is a general lecture, I thought there might be some students here who have a career in mind. The value of the non-root crops in the world, according to the FAO, rounds up to about $1 trillion per year. And these are of the shoot apical meristem derived crops. I excluded the root crops from this figure. And so the people sell shoot apical meristems in their products for $1 trillion every year all around the world. There are about 5 billion metric tons of major non-root crop plants per year, so that's 5 trillion kilograms, 10 trillion pounds of shoot apical meristems are on the market every year. In 2007, according to Wikipedia, which is the ultimate source of all knowledge, about 1/3 of the world's workers were employed in agriculture, and about 20 years ago, this was more than half. And so from the dawn of human civilization until about 20 years ago, the majority occupation of the average human being was being a servant for shoot apical meristems. And so from that, you can see that it is the most important thing in the world. It's what all of mankind and womankind have devoted themselves to for all of human history, is making these little things happy. And so there can't be anything more important. If you look at the entire world biomass, and I've gone to an even greater authority than Wikipedia here, Google. There are supposed to be 1.3 trillion metric tons of plant biomass on land in the world, all except the roots, the products of shoot apical meristems. And so this is the dominant object in our universe on Earth. It's the only thing that matters. If all the animals disappeared, it wouldn't make a lick of difference to the shoot apical meristems, except for the ones that are, crop plants. Somebody be hoeing the ground for them, and the ones that need insect pollination. But if all the plants disappeared, the animals wouldn't be here for very long, nor would anything else. So that's what I mean. We really have to understand how this thing works if we want to serve ourselves with food and fiber, medications, natural landscapes, ecosystem services, anything you want to say. We'd better figure out how this thing works. We don't know diddly about how this thing works. All right. One other thing I want to point out, and I just flat out pirated this picture off the internet. So take it off the screen if this is copyrighted, because I can't remember where I got it. It's a piece of thread in the eye of a needle, and I superimposed that shoot apical meristem picture at about the right size on that. So this thing, 20 of them would fit through the eye of a needle. And yet 1.2 trillion metric tons of stuff that results from them is sitting out on the Earth. And here's a picture, just a optical section where the nuclei are glowing in this embryo of Arabidopsis. There's the cotyledons, there's the root and the root apical meristem, which I'm belittling, and there's the shoot apical meristem, which in the end will give rise to everything in the plant that you don't already see here. That is, everything above the cotyledons will derive from this population of stem cells. So it has a series of remarkable properties. It's even more complicated than I've shown so far, and there's an important human imperative to understand how it works so that we can rationally grow the plants that we need. So here's a series of questions that have come up over time in my laboratory that relate to how the shoot apical meristem works. And I'm not going to answer any of them because you don't know the answers. I'm not going to go through all of them at this talk, but I'm going to try and say a few things about cell number control, about the regulation of gene expression domains, and if there's time at the end, and I'll keep an eye on the time, at the rate I'm going there won't be, I'll talk about regeneration, because plant scientists have already solved all the problems that the entire human stem cell world is working on, which is we already know how to take any cell, make it into a stem cell, take that stem cell and regenerate it back into an entire plant or any organ we want. And so the problems that the medical world are struggling with at such vast scale are ones that have been solved a long time ago for plants, and I'd like to talk a little bit about that as well. So cell number control. We have this problem. Here's a shoot apical meristem early in the life of an Arabidopsis plant, and here's one late in the life. At this point, it's making leaves, and these are the primordia of leaves. And at this point, it's making flowers, and these are the primordia of original flowers as they form. And what you can see is that the size of the shoot apical meristem early in the life of the plant and later in the life of the plant is about the same number of stem cells. The implication of this is that as cells divide in the shoot apical meristem and they leave the shoot apical meristem to become parts of leaves or flowers or to become the cells of the stem beneath the shoot apical meristem, there are ones that depart to differentiate are exactly replaced by the ones that divide in the meristem. Because if they were even off by a few cells a day, This thing would, if it was too few, this thing would disappear over the course of the life of the plant, which it doesn't. If there were too many, it would get bigger. And so the cell divisions are exactly balanced against cell departures. And if you imagine this for something like a redwood, which does this for 1,000 years, there can't even be an extra cell division a week, or that meristem would be enormous. So we approached this problem first a little more than 15 years ago, and we thought, well, if this balance is so perfect, we should be able to disrupt it by doing a mutagenesis, that is, putting the seeds in some chemical mutagen, changing the DNA, growing the plants up, and seeing when we eliminate the function of particular individual genes, whether we can get meristems that get bigger or get smaller through the life of the plant, rather than maintaining this delicate balance between cell departure and cell division. And so one of the mutants we got is one called clavata3. We didn't originally get it. Other people found it before we rediscovered it. And there are other mutants, all called clavata, which have the effect that when the genes are knocked out, there's a normal shoot apical meristem. You can't see it. It's too small to see. But there's the shoot apical meristem in this. They get bigger through the life of the plant. So this balance of cell division and cell departure is disrupted in favor of extra divisions compared to departure of the cells to differentiate. And there's a scanning electron micrograph at the same magnification of the shoot apical meristem normal. And there's the mutant one, which isn't such a smooth, nice-looking structure. It still makes the flowers and leaves around it, but it's vastly overgrown by this late in the life of the plant. And so using this, we wanted to get some key to the mechanism by which the cells decided how frequently they should divide. You've seen these before. This isn't a mutant. These meristems sometimes don't maintain their perfect balance, and they overgrow, and they continue getting larger relative to what they should be doing, and they make these crested certs of plants, which you can find in the Arizona desert by looking down, and photographers have made tracks to them. And so we collected a couple of these genes, a number of these genes, and I just want to tell you about two initially, and maybe I'll bring in a third gene to give you an idea for how this work was done 10 years ago or 8 years ago, and what we learned from doing it. So that we cloned the genes, that is, we got the DNA sequences out of these genes by methods that would be easier today than they were then, and we found that the clavata3 gene codes for an extracellular protein. It gets spit out of plant cells, and when it's out of the plant cell, it gets cleaved down, as has been learned later, into a 13 amino acid peptide, which sits outside the cells and is a signaling molecule. It moves between the cells in the shoot apical meristem. And one of the other genes was a transmembrane receptor kinase, that is a protein that sits in the membrane of some of the cells of the meristem, binds to this clavata3 peptide in the extracellular domain, and when it does that, it causes a chemical reaction in the cytoplasm of the cell that signals to other proteins and sets some process in motion. So there's a transmembrane receptor kinase, clavata1, and a ligand for the transmembrane receptor kinase, clavata3. And we look where the RNAs for these genes are made in the meristem. These are sections of the meristem stained for the position of these specific RNAs. These are floral meristems. That the clavata3, the signaling peptide that's exported from the cells, is made in these ultimate stem cells at the very tip of the shoot apical meristem, from which eventually the peripheral cells that make organs and the rib meristem down in the middle here that makes the stem derive. And clavata1 is in a population of cells just beneath it. So what we reveal by this mutagenesis and doing the standard sort of developmental genetics is that there's a signaling system in the meristem where there's a signal made in the very tip of the meristem that goes down to the cells beneath it, and if you disrupt this signal, something makes the meristem get a lot bigger. So we've got the beginning of our inference of a mechanism for cell number control. And I also want to point out that another thing that this shows is that all the cells in the meristem are not the same. The ones at the very tip express at least one different gene from the ones beneath them. And so that although the cells look homogeneous, they're not homogeneous. They're different from each other. And as the cells divide and they pour out, as a cell divides here, if it leaves this region, it turns off this clavata3 gene. And if it happens to divide here and enter this region, it will turn on the clavata1 gene. So any individual cell goes through a series of stages of gene expression as it pours through the meristem, while the domains of gene expression remain constant. So the cells move, but gene expression is forever. In fact, given that plants that are diverged from Arabidopsis by 150 million years have exactly these same patterns of expression, you can say that these pattern domains of gene expression move within the meristem probably millions of times more slowly than the continents move across the oceans. They're very stable over a very long, 60 million years ago, all the continents were in one. And they've moved all the way across the Pacific Ocean since then. This stuff hasn't even moved one cell diameter. If we look at the mutants using another technology for looking at the expression of these genes, or the pink here is the expression of the clavata3 gene, it's on, and this is a clavata1 mutant, so we knock out the mutant, we have these giant meristems, and this domain has gotten a lot bigger. So the domain where clavata3 is expressed is enlarged, and it's equally true in a clavata3 mutant or a clavata1 mutant that the domain of clavata1 has gotten a lot bigger. So this now allows us to infer a mechanism for what's going on in the meristem at a certain level, and it's this. There are these cells that are the stem cells or the cells underneath them, the rib meristem cells. These two cell populations maintain the same relative size, and the way they maintain their same relative size is with a signaling system. We've just shown this part of it. That is, these cells spit out something that activates a kinase in these cells, which tell these cells, this domain, not to get so big. And so it represses the size of this domain. If you knock this out, this domain gets bigger. But actually, when you knock it out, this domain also gets bigger. So there must be a positive signal from these cells that goes back to these cells above them and tells them to grow. So we can infer from developmental genetics this little mechanism whereby these two regions remain in appropriate size because these ones tell these ones not to, that tells this domain not to be so big, and this domain, when it gets big, tells this domain to get bigger. If it gets small, tells this domain to get smaller. So it keeps the two domains locked in the same relative size. So it's part of the overall cell control mechanism. And we can infer this, and we can demonstrate it from a series of genetic experiments. For example, if we create a transgenic plant that makes far too much of the signaling molecule clavata3, that should repress the growth of the cells underneath it, and the meristem should go away, which it does. If we overexpress it, plants look like a mown lawn. You don't see any new leaves still being made there. And so one prediction of this model was what would happen if we overexpress the gene, and that prediction comes out. Similarly, if it's true that this signaling molecule works through the transmembrane receptor kinase, if we knock out the transmembrane receptor kinase, it shouldn't matter if we overexpress the signaling molecule because the signal can't be received. And that's what this experiment showed some years back, maybe 10 years ago, that if you overexpress the signaling molecule in a background of a loss of function of the transmembrane kinase, nothing happens. You just get the bigger meristem. So this is the way developmental genetics is done, step by step, looking at phenotypes after mutagenesis and inferring mechanisms from looking at what's going on. And so what we have here is I've added some extra pieces here. This picture from a recent thesis of one of my students, but we have clavata3, clavata1. There's a transcription factor called Wuschel, which is controlled by clavata1, which is involved in this circuit and signals back up to clavata3. So we have this peptide that signals the receptor kinase. And if, for example, we start to overexpress Wuschel, the clavata3 domain gets bigger. But what experiments like this don't tell us is the mechanism by which it gets bigger. And you can think of three mechanisms by which it gets bigger. One is that these cells just start to divide faster. Another is that every time a cell on the boundary of this domain divides, normally one daughter cell would turn off the gene and the other would keep it on, and that's how it maintains its size. But suppose they both keep it on. So it could be that you don't get the asymmetric cell division that you would normally get. And a third possibility, one that didn't occur to us until we did the experiment, was that it could actually be that it's these cells that are affected. And that what happens is that when you activate Wuschel or overactivate clavata1, underactivate clavata1, you cut off clavata1 because clavata1 represses Wuschel, then these cells that are outside this domain become stem cells. And they differentiate backwards from these multipotent stem cells that are here to make leaves and flowers in the peripheral zone, and that they may turn into the pluripotent stem cells that can make any above ground part of the plant. something you don't often find and people don't think about in animal development, but they should start. Because, okay, so now we apply our new way of doing things. This is as far as we could get with traditional developmental genetics. We got a lot more mutants. We studied this in a lot of ways, the biochemistry and stuff. It didn't teach us any more about the integrative mechanisms of the plant. So let's introduce our new toolkit now of computational morphodynamics. The first part of it is live imaging and image analysis. You'll note that all of the conclusions we derived in the earlier part of this work that I've talked about so far was from plants that are dead. So we have no idea the rate at which cells are dividing, the rate at which domains are changing, the rate at which the cells are moving through the meristem. All of that information is lost doing these things in a traditional way. So we began by setting up a confocal microscope and some fluorescent reporters for gene expression and starting to look at all these things when the plants were alive. And it was eye-opening. The second thing is that once you start to do this, you get a lot of information about what's going on in a plant. Enough information that I, at least, can't any longer use my intuition to infer models because there are too many things going on. And so we had to bring in a bunch of mathematicians and computer scientists who could make these, our hypotheses, explicit and run them with all their feedbacks in a computer and then see if they're plausible. If we can get it to work in a computer, it's at least plausible. If you can't get it to work in a computer, you probably should think again. If you do get it to work in a computer and you cast the model correctly, such that the parameters in your equations are things that you can change in an experiment, you can then go back and test your model by changing those things experimentally, and you should get the same result in the plant that you get in the computer. And if you don't, you know you've got something wrong, and you can refine your hypothesis. So you then iterate between live imaging, computational modeling, which is a type of explicit hypothesis, a computational model, and then testing the hypothesis and then refinement of the hypothesis if it turns out it's wrong. Of course, you can never prove the model's right. You never can do that in science. All you can do is keep trying to prove it's wrong and failing, which I probably shouldn't say if there are undergraduates in the audience, because that really is the height of your scientific life. If you want to go into life of science, the best you'll ever be able to do is keep trying to prove your own hypotheses wrong and failing. And if you succeed in doing it, it's worse. And so this is what we call computational morphodynamics, an explicit computational part and live imaging with a lot of attendant image processing and image analysis. So here's a meristem as viewed through the lens of computational morphodynamics. What we've labeled here is only the plasma membranes, and here are the flower primordia forming. Around this meristem is all these cells in the ultimate stem cell population that expresses clavata3 or dividing. And from this sort of information, you can get a complete clonal analysis. You can get the rates of cell division anywhere on the meristem in mutant or in wild-type plants. You can fluorescently label the expression domain of any gene in which you're interested, as we've done here for a couple of genes, one called revoluta, which is a nuclear protein, and one called PIN1, just as an exemplar. It's a plasma membrane protein, and this is not a time lapse. We're just showing that we have three-dimensional images, and we're looking at different floral primordia from early ones to later ones where a revoluta starts to be expressed very shortly after the primordia are established. And so you can look at the domains of gene expression by having fluorescent adducts to the proteins and then putting these genes back into the plant, letting them express and serve their normal function. But in addition to the normal function of the gene, they also glow because of a trick that you, the molecular biologists, have used. And so with these live imaging methods, we're able to approach our questions of clavata3 and clavata1. And the question I'll take you to the answer of is exactly what's happening to the central zone when, that is the region where clavata3 is expressed. Here's how we look at the meristem from the top down, and there's our stem cells. But we can reconstruct computationally this picture in 3Ds and look from the side at the clavata3 zone. Similarly, there's Wuschel, which is co-expressed with clavata1. If you label clavata1 with GFP, you don't see anything. I'll come back to that. People have tried. You don't see a thing. There actually isn't any clavata1. There isn't enough clavata1 protein to be able to see it in a wild-type plant. So there's Wuschel for our rib meristem. You can see if we reconstruct it from the side, it's down there in the core, and this happens to be a peripheral zone marker. And so we can do an experiment then where, first of all, we can look at our familiar clavata3 mutant. We've labeled the membranes here with a lipophilic dye. Clavata3 is in green. And you can see that the clavata3 domain is much larger, as I showed before, in a clavata3 mutant when the meristem gets larger. And so then the question becomes, what's the mechanism by which it grows larger? We need to add some dynamism to the genetics as well as to the imaging to look at this. And so that we built a system. This is Venu Reddy, a postdoc in the lab some years back. He's at Riverside now. That built an inducible system where he could put a human drug, dexamethasone, on the plant, and it would cause the clavata3 gene to turn off and stop signaling clavata1. It works like this for the aficionados in the audience. Well, the aficionados already know. That is the animal nuclear hormone receptor that binds to glucocorticoid, the rat receptor, has a steroid binding domain that holds proteins in the cytoplasm inactive. When it binds the steroid, it transits into the nucleus and activates the protein. So we attach this to an artificial transcription factor, which could activate an RNAi complementary to the clavata3 gene. And so when we put dexamethasone on, this transcription factor activates our RNAi gene, which then represses the clavata3 gene. There's some feedback there too. And so if we do that, we drop at 0 days, we drop our drug on 6 days later, our central zone has enlarged. If we look at the time course on the way through that, we can watch the domain enlarging. And what you actually see is if you look at individual cells, you see that the rate of cell division is not fast enough to create this increase now that we know how fast it is. That, in fact, these cells divide on average only about every 96 hours in the central zone. And so this whole experiment, each of these cells is divided on average once, but these particular cells that you identify have become encompassed by the clavata3 expression domain. In other words, the peripheral zone cells, the multipotent stem cells, are getting respecified to be the less specialized pluripotent stem cells as a result of the signal from the rib meristem. And so by applying these live imaging methods and getting some rates in there, you can begin to answer mechanistic questions that you can't approach with traditional developmental genetics. Here's another example where, in this case, we activated the Wuschel gene, which does the same as inactivating the clavata3 gene, causes this domain to expand, and you can see that it encompasses cells that aren't dividing in the course of the experiment. What's the next step? I promised you computational modeling, and I'm not going to say too much about this, just to show you that it works, so that if we take all these feedbacks that I've just given you, a clavata1-3 complex, when you get the clavata3 protein, the clavata3 gene, it signals back to Wuschel in particular ways. And if we build this, we can reproduce the expression domains of these genes in the computer, and that's a little bit phony. I'll show you the equations for that in a second. We set it up so that the expression domains would be right. The model doesn't explain that. But once we activate clavata1 and clavata3 and these rules for the expansion of the domains at appropriate rates, we can reproduce the behavior that we see in the shoot apical meristem in a computer. Another one for the aficionados. This is the beginning of that model, the activator model. And some of this, over here, this is just, this part's bogus. It's just to make sure the genes are in the right place, these two equations. And this encompasses the Wuschel, change in Wuschel concentration with time, and the change in another signal with time that's proportional to clavata3. Diffusion of clavata3 through the meristem, you can see the diffusion equation. And so if we apply that here, for example, is an experiment that was done by Reinhardt and his colleagues in 2003, in which they took a laser and drilled a hole right through a shoot apical meristem and then looked at by in situ hybridization in dead tissue at what happens to the Wuschel domain after you do this. And our model exactly reproduced the experimental results. That is, what happens is you reestablish a new Wuschel domain, bifurcated. And there's the actual experimental result, and this is what our model did, as done by Henrik Janssen, our collaborator. And so you begin to reproduce computationally these feedbacks, and the more realistic the model is, the more it suggests to you in the way of experiments. I want to show for the people who are into shoot apical meristems one reasonably new result. What we're showing here is clavata1 expressed, in this case, from a promoter that makes it express everywhere in the shoot apical meristem, not just the rib zone. And if you have it in a wild type background and you keep the plant in the dark so the vacuoles don't do much degradation, and you turn the confocal microscope all the way up, you can see some clavata3 in degradative vacuoles. in a clavata mutant background, it's a plasma membrane protein, just like you'd expect it to be. That is, in the presence of the ligand, clavata-1 is internalized by endocytosis and degraded, which is why you can't typically see it in a GFP experiment. And here's a close-up of mutant and wild type with clavata-3 on it, or clavata-1 on its own promoter. So there it is in the presence of the ligand. There it is in the absence of the ligand. So it's sitting there on the plasma membrane ready to catch this signal, but as soon as it catches the signal, it's degraded. I said we faked in that model the domain of expression of genes like clavata-3 and Wuschel and clavata-1. We just put them in the right place in the meristem with that model by using a couple of tricks. Epidermally secreted activator and another activator down on the base of the stem, which were the Bruselator equations at the bottom there that you saw. How does it really work? How do these domains of gene expression maintain themselves constant as the cells pour through them? And I want to say just a couple of things about some recent work that, again, our live imaging has led us to. So we have this question, how is it that that gets expressed in the right place and that stays in the right place, even though the cells that are expressing it are moving top to bottom right through these zones? There's some observations in literature that if you put a plant hormone called cytokinin on a shoot apical meristem, it gets bigger, just as if you had knocked down clavata-1 expression. In fact, my graduate student, Sean Gordon, now postdoc, showed that if you add cytokinin to the meristems and measure the amount of RNA for these various genes, this is one involved in cytokinin signaling. This is Wuschel, the thing that's on the clavata-1 domain, and clavata-1 itself. You get a gradual reduction in the amount of this receptor. That is, the hormone represses the receptor, activates the gene for the transcription factor at very high concentrations. So you get a complex dynamics as a result of this plant hormone when you put it on in large quantities. If you look at the actual signaling of the hormone in the shoot apical meristem, and again we do live imaging using a fluorescent reporter that we got in this case from Jen Sheen's lab, PTCS, which is an artificial promoter that activates the attached fluorescent protein when it experiences this hormone. So it's an indicator of hormone concentration. You can see that the expression domain of the place where cells are responding to cytokinin in the meristem is coincident with the Wuschel and clavata-1 domain. So there's a clue that there's a hormone that activates genes in the exact same place as these. If you look at the receptor for the hormone, it's exactly co-expressed with Wuschel. The AHK4 is one of the receptors for this particular hormone. And so that you have a series of feedbacks here. The hormone induces Wuschel, it represses clavata-1. It actually activates, Wuschel is known to activate, it inhibits repressors of cytokinin signaling. So when Wuschel is on, it makes cytokinin signaling occur more easily. And yet the Wuschel gene itself is responding to cytokinin. So you have a series of feedbacks going on in this meristem that, again, you can't encompass in your head. And so, and here's also, if we add cytokinin, what we can see is this Wuschel domain gets bigger when we have more cytokinin, just the same way as the clavata-3 domain did. It incorporates cells that used to be outside the rib meristem and activates the rib meristem genes in them. So the rib zone gets bigger as a result of hormone treatment because cells are getting respecified from regions where they're more differentiated to regions where they're less differentiated. So here's our network with all these feedbacks, the series of equations that represent that. And when we do this, we can actually get back some quite realistic behavior, which depends on the assumption that there's a gradient of the hormone from the top to the bottom of the shoot apical meristem. And we're now looking at the biosynthetic genes, and it's already known that some of the biosynthetic genes for the hormone are expressed strongly at the top of the meristem, so that there could be a gradient of cytokinin. Of course, we've just pushed the problem back a step, which is we wanted to know how Wuschel was expressed in the middle of the meristem, and we've pushed it back to now we need to know why it is that cytokinin is only synthesized at the tip of the meristem. But you know, it's a life cycle, and so if we push it back far enough, we'll come all the way around and we'll be good. And so it's not an insoluble problem. And here's some of the results of that computational model showing things that were then verified by observations. I'm going to skip these because I'm talking about this tomorrow, and anybody who wants to hear about this should come and hear me talk about how it is these flowers get put out in such a lovely geometric pattern. And that's what the subject of tomorrow's lecture is going to be. And this phylotactic pattern, which is an old problem that I think we've solved. But I want to finish up by talking just a little bit about regeneration. One more miraculous property of these amazing shoot apical meristems. So think about what we're all hoping will happen with human stem cells, which is that people will learn to take any cell in your body and get it to dedifferentiate into a stem cell, and then use that to grow any type of organ we want and create replacement organs for yourself. Be a nice thing to be able to do. You know, that there comes a time when we all need a new liver or lung or kidney or something, and it would be good to be able to do it. Plants, amazingly enough, do this much more easily than animal cells, and it's been known for a long time that they do. I mean, you've done it yourself. You pull up a dandelion in your lawn, if you leave just that much of the root, you get the whole plant back. So you know that a root can regenerate into a shoot. Or if you take a rose cutting and you dip it in root tone, which is a hormone, auxin, and a fungicide, and then stick that in the ground, that shoot will make new roots, and you get a rose bush. It'll have cruddy roots because normally they graft them onto something that's resistant to the crud that lives in your soil. But it's we know that plants can do this stuff. Or if you look at a kalanchoe, you know, there's leaves. You have these leaves, and then there are these little plantlets that form on the edge of the leaves, little roots, and all, and they fall off, and they become new plants. So we know that this sort of stem cell regeneration is a solved problem in the world of botany, even though it's one of the great frontiers in the world of animal science. Oddly enough, as far as I know, nobody in the world of animal science has read the plant literature to see how this actually works, but I'm sure they'll get around to it eventually. And so here's a picture of this happening, that what you do is that you can take any tissue in the plant, a petal, a cotyledon, a root, mince it up with a razor blade, and throw it in an appropriate culture medium, which has both cytokinin and another hormone, auxin. And then it'll grow up into these masses that are called callus, these big cell masses. They're about the size of your thumbnail, each of these. And then if you change the culture medium to have a different ratio of auxin to cytokinin, somehow spontaneously new shoot apical meristems form in this stuff, and they can be from roots, so they never had a shoot apical meristem ever. But they self-organize these complex structures with all their domains of gene expression, and they grow normally, and they make leaves and flowers and shoots, and they're fine. And so no one really had any idea what was going on to make this happen. It's amazing, but no one had ever looked at it before. But you can look at it with live imaging. You can use our methods of computational morphodynamics, and you can put your microscope on a piece of callus, and you can induce it to make new shoot apical meristems, which happens in here and there, various places, and you just run the movie backwards after it happens, and you can see the earliest events that are occurring in those cells that later will be the new shoot apical meristem. And so you can add, again, a dynamic point. And in this respect, you can also say which cells it is that later will give rise to the shoot apical meristem, because you can run time backwards in a movie. And so here's the process, and these various media. The callus-inducing medium makes this callus. If you put it in shoot-inducing medium, it'll make shoots. If you put it in root-inducing medium, it'll make roots. And here it is, what happens in the very earliest stages of shoot apical meristem regeneration occur. We're looking here at three different things. The blue is just chlorophyll fluorescence, so we're seeing where the tissue is, where the chloroplasts are. Pin 1, which is a transporter for a hormone, auxin, which I'll talk about tomorrow, is in green, and that's the first thing that happens. And another gene, revoluta, which is the one I showed you before, in red, also turns on right there, which is a meristem marker. So the first thing that seems to happen is a transporter for a plant hormone called auxin, whose concentration is important in these media, so we know it's involved in the process, come on. And this transporter moves the auxin towards a peak. The gene gets activated maybe at random by random fluctuations in auxin concentration, and this gene itself is auxin-inducible, so there's a positive feedback that brings it to a high level. It then directs auxin flow toward a point, and it's tomorrow's talk is when I'll tell you why it does that. And so it creates a big peak of auxin concentration, which then can induce the revoluta gene, which then leads to a series of subsequent processes, and eventually you get this whole complex meristem formed here, And these are new leaf primordia being formed around what's now a new shoot apical meristem, and Rev being expressed in the early developing flowers. So we can go back and look at the series of gene activations that give rise to these new shoot apical meristems and allow these very complicated structures to self-organize and come up with one gene after the other, after the other, after the other, until we, with all these multiply marked fluorescent strains, until we can come up with a coherent picture of the patterns of gene expression and patterns of protein movement at a subcellular level of resolution that are leading us to the de novo formation of shoot apical meristems. I said before that a meristem is a lot more complicated than a 777 aircraft. Each cell in it is more complicated. I had a colleague, the reason I say 777 is I have a colleague in engineering named John Doyle, and he once came to talk to me about biology, and he was struck by the fact that a 777 aircraft, which he works on the computer-aided design of, had the same number of working assemblies as a yeast cell, which is about 100,000. There's 100,000 assemblies of proteins in a yeast cell, maybe. And similarly, there are 100,000 parts that Boeing buys and puts together to make a 777. They buy individual rivets, and so they count. They also buy engines from GE, and so an engine's also an individual part in those programs. And so it's about comparable. And so he said, if we can encompass the structure and the function of a 777 aircraft in my computer programs, why can't we do that for a yeast cell? And my answer to him was that there's some differences. One is that the rivets don't move in a 777, while the proteins move and associate with each other in a lot of different ways. And so there's a problem of mobility. Secondly, a yeast cell finds its own fuel, and your 777 doesn't. And finally, the yeast cell reproduces. So if you can make me a 777 that found its own kerosene and split itself when you needed a new airplane, then we'd be talking about something at the same level of complexity. Let me add a third thing that we've now realized. This years ago conversation with Doyle, I wasn't thinking about it. But I should have also added that this thing will self-organize, so that you should be able to take a shovel and a bunch of metal ore and pile it up in the middle of a runway and wait a few days, and it should turn itself into a 777. Then you're talking about something like a yeast cell or a shoot apical meristem. So what happens here? Here's a summary of some of the things that happen in which this pin 1 gets activated. It starts to concentrate auxin in a peak. It induces some genes like WUSCHEL and STM, which are involved in cytokinin biosynthesis. So you end up with a particular pattern of auxin and cytokinin around these little points where, probably for random reasons, the pin 1 protein started to be expressed. Again, we can express this as a somewhat theoretical gene regulatory network, and this is the way most geneticists would represent such a network that also includes hormones and synthesis of cytokinin. And each of the steps in this is pretty much validated by biochemical or genetic experiments. But you can't say what a network like this is going to do by staring at it. There are too many feedbacks. But you can tell your computer. And when you tell the computer about this and you have it play out on a two-dimensional plane of cells, what you get are these little sharp auxin peaks, which sit in a background of shallower cytokinin peaks that make bullseyes, where there's high auxin surrounded by a moderate level of cytokinin, which then can induce the appropriate genes to create the radial structure of the shoot apical meristem. So it self-organizes one step at a time, and we can reproduce that in a computer. So I think we begin to understand how this sort of self-organization can work. I think I've talked about long enough. I want to say one more thing, just for a little bit of fun, which is a recent discovery. That work I just showed you was done mostly by Sean Gordon in the lab and Vijay Chikramani, who's one of our computational guys. And in my lab, they're both computational biologists and biologists. And as I'll point out at the end, a very large set of collaborators all over the world. One other thing, you might ask yourself about the formation of this callus. The literature of plant biology tells us that what happens when you mince up a root or a petal or a cotyledon and you let it form into this callus in callus-inducing medium is that it's a de-differentiation process, and that the cells become undifferentiated and then they become redifferentiated to form the new shoots. Actually, it turns out that's not it at all. And things are a little bit more complicated than anyone thought. And what it is that the callus, whether it's derived from a root or derived from a petal, is a root. and expresses the genes the root expresses. It expresses them in a highly patterned way, just like a new lateral root primordium. And if you have mutations that prevent the formation of new lateral roots, you cannot get the plant to form callus. So it's a process of root formation, even from above ground organisms. And so when we make new shoots, we can take a petal and we mince it up and we make callus. We're actually making that petal cells, certain set of cells, the phloem parenchyma cells in the petal, grow up into a mass that forms a new root. And then we're inducing that root to make a new shoot meristem. And so we have another set of processes to study now on the way to making callus. And the way we got into this was because we wondered which cells and what the cells were doing in the callus before they started to make the shoot apical meristem. And it turns out what they're doing is being a root epidermis. And so this is Kaoru Sugimoto's work, a new student in the lab. And so I've gone through a postdoc in the lab, and that's will be published one of these days. And so I've talked a little bit about cell number control and use that to give you an idea how this new computational morphodynamics approach gives you the ability to get more mechanistic information than you could get the previous way of doing developmental genetics. And so I would recommend to everybody some live imaging and some computational modeling. I talked a little bit about gene expression domain regulation and how that involves cytokinin, a plant hormone that's been known for a long time. And then I skipped these for tomorrow. And then I talked a little bit about this amazing regeneration. And even after all the years that it's been studied in plants, there's still some new things to learn, like what the nature of callus is and what the earliest events are in this remarkable self-organization of the complex shoot apical meristem structure. And so with that, I want to show you the people in my lab just to show you that it's a great group. And I didn't get to talk about all of their work. There's Kaoru, whose work I was just talking about a minute ago. There's Sean, whose work I mentioned. There's Vijay, who I showed some of his computational models. And all the other people are doing things. And Paul is looking at cytokinin biosynthesis. Some of the other people's work, and Zach did that work on Clavata-1 internalization, Zach Nimchuk. And I'll talk about the work of some of the others tomorrow. I didn't get through all of it today. I also want to point out that all this work has depended on a very long-term collaboration with a mathematician, computer scientist, physicist, and overall genius at the University of California, Irvine, named Eric Miolsnes, whose daughter is an undergraduate here. I don't know if he persuaded. There she is. There's Claire. So her dad has been for 20 years my collaborator in doing all the mathematical parts of this and patiently explaining to me over and over again what is a differential equation and what that means. A former postdoc who worked with me, who's now in the Department of Theoretical Physics at Lund University, Henrik Janssen, who's been much involved in all this modeling. His postdoc, who used to be a postdoc in Eric's lab and then moved over to Henrik's lab, Pavel Krupinski, most of his models I'll talk about tomorrow. Bruce Shapiro, somebody Eric and I hired many years ago, who's been at the Jet Propulsion Lab in Caltech ever since, and has been doing the computational modeling with us. It takes a team. Jan Tross, a collaborator in France, with whom we've done a bunch of stuff, but mostly the stuff I'll talk about tomorrow. And we could go on, I will go on tomorrow, with even more and more people all around the world who are collaborating to do all this. It's not easily done in a single, with a single lab. One hopes that the students of the future will be able to do this all themselves. And my students now, some of them can, but I can't. So I need collaborators. This is another view of a meristem, just to let you watch it grow while I answer questions. Thank you.
Two questions. One more further than the other. Doesn't actually your last data slide actually prove that the root apical meristem is more important than the apical one because it comes first? Secondly,
I won't answer that, Richard.
secondly, though,
In case it gets back to Ben Sherez or Liam Dolan or one of these guys who I've been razzing about working on roots for 20 years.
Secondly, though, as you begin to understand and dissect out the process of regeneration, you glibly say that we can do that. In fact, it's actually taken years and years of empirical experimentation to get plants to regenerate. And within a species, there's this famous correlation between, or inverse correlation between economic importance and your ability to regenerate plants. And also, though, there are different species, some of which you still can't regenerate. So have you been able to use this information, or do you think you will be able to use this information to get more efficient regeneration protocols?
I think we probably will be able to. I think we could already. We haven't tried. But I didn't mention some of the very earliest steps. So what happens after this callus forms and it reaches its root-like state, which is easy to assay, then the cells on its surface partition themselves in shoot-inducing medium into domains of expression of either Wuschel or cup-shaped cotyledons too. I forget which. I think it's the Wuschel domain. It's the only one that has a shoot-apical meristem. And the next thing that has to happen is pin 1 has to go on at a particular point. There's no reason why you couldn't look at all these things in any plant and make sure they were happening step by step. So you could very quickly optimize the activation of the root expression program to form this type of callus. There may be other types, we don't know. And then once you have that, you could optimize the culture conditions over a very broad range of conditions and look for activation of Wuschel or cup-shaped cotyledons too, if you could do it, rather than waiting to see if shoots formed, and so that you could speed the assays up many, many-fold. We haven't done it, but I think you could.
Intra-specifically, you could test your hypothesis by looking at genotypes that do regenerate and those that don't easily. And you should see a differential gene expression pattern of the key ends of the key
Absolutely
proteins.
should, as we do when we have, say, our ALF4 mutants, which don't make lateral root primordia and also will not form callus, which is the earliest mutant that we have, yeah.
But there's no genetic variation in Arabidopsis fortis.
Well, that is ALF4 versus...
Yeah, natural genetic variation.
It's natural.
Who needs natural variation if you've got the real thing, which is the stuff you induce in the lab?
Now he's quoting you too.
Yeah, that's right. Now he's going to give me more stuff about the Hadean parts of the plant. Yeah, Siobhan.
So when you're modeling the gene expression domains and how they're formed, like the one of Wuschel's, there's a lot of different components that all interact with each other. Have you been able to figure out how robust each of those components are to small fluctuations? And are there any components that need to be more tightly controlled than others to set up them?
The first question is, yeah, we've looked at the robustness in the living material, as have others. We cut the stuff in half, laser drill it, ablate cells.
Opposed to the sledgehammer kind of, modifications, have you played around with subtle changes? And
We haven't seen that anything changes when we do anything subtle. You have to hit it to change anything. It is very robust. And then the second answer is that we've also done a robustness analysis in the computational models, and they also seem to be very robust to wide parameter changes.
There's no one component that needs to be more tightly controlled.
Oh, things don't work that way. The whole thing is a component, all interacting, all with feedbacks to each other. So if you change one thing, you change everything, which is why you need the computer. And why you can't do it the old way, thinking about it one gene at a time. If you knock out completely the activity of one gene, the meristem changes, but everything about the meristem is changed. The whole network is different, and so you need the computational model to look at that. And the robustness comes from the interaction of the parts. None of the parts are that important. You can take them all out, and the meristem, is okay for a while. You take Wuschel out, it kind of goes away. But it's the interactions, not, yeah.
I was going to comment on that in deference to Liam Dolan as well, that the roots seem to be more important. But my question is, you seem to have been initially somewhat surprised that you had induced pluripotency. Wouldn't that be intuitively expected when you damaged the meristem in the adjoining cells?
I don't know, I didn't.
You didn't.
What can I say? I've been a little more on top of things. I probably would have, but
It's just when you think of removal.
Probably science like this isn't much fun for people who are really smart because they figure all of it out in advance. But for somebody like me, it's like a continual process of discovery. When I get out of bed in the morning, it all looks new to me.
I was just referring to, when you remove apical dominance, you're going to get it coming.
No, but when you're removing apical dominance, you're not. No, the meristem is already there. It's already fully formed, just sitting there, and you're releasing it. But it's all there already. You're not making anything more pluripotent. Somebody else must have a question.
After that smackdown. I had other questions, but I won't ask them.
Oh, go ahead. Come on. You can take it, and I can take it.
Root epidermis, but in your picture it was scarecrow. Scarecrow's not in the epidermis of the root.
No, we weren't looking at the epidermis there. We were looking at the confocal section right through the thing. And you get the whole layer. You get endodermal markers, cortical markers, pericycle markers, steel markers, all from outside to inside in the callus. It's amazingly organized. I mean, it's kind of bloated, but it's organized. There are QC markers that are kind of all over. That's a little disorganized.
So the layered structure
It's still there. And in fact, you get stripes of hair cell and non-hair cell things on the epidermis. It's really just like a root, which kind of scared us. Yeah, all right. So the root apical meristem is more important. Yeah.
So once a root apex sort of organizes, then what part of it, or is that region itself going to reorganize into a shoot apex then, or?
We don't know. That's the next step.
Okay.
That's why we got into looking at what's going on in the callus, is so we could find out what events might exist before we see the new shoot apical meristems occurring. And we know there's some events prior to that because of the Wishow-Cook-2 partitioning, in which the meristems only form in one of those domains. And it was that cytokine and auxin model in which we were really trying to look at the Wishow-Cook-2 partitioning. But that's what the lower right figure was, with the colors were those different genes being expressed according to that this pretty hypothetical model of gene interactions that we have there. But you got to try. It's more dishonest just to have the arrows and bars.
So a wild thought, I mean, is it possible that some structures organize into roots because of high auxin and, like stochastic differences in how the auxin itself is distributed across the callus?
Yeah.
And that
could be.
the places that organize into shoots are, maybe auxin depletion is involved in that because you pull stuff away.
Actually, it seems to be auxin concentration that makes the shoots, but we haven't looked at the root meristems. We could go back and look at root meristems forming too, but nobody in the lab has done it. All this regeneration work has been done entirely by 3 people. Sean, as a graduate student, Kaoru looking at the callus, and Vijay on the modeling. That's it. I wish I could get more people in the lab interested in it, but you know how it is trying to persuade a new graduate student to do a hard project. Yeah.
I was wondering, have you looked at embryogenesis? Because there are many calluses that, just...
Yeah.
And then you have a very interesting structure.
That's correct. No, we haven't. And in fact, people have shown that clavata mutants undergo somatic embryogenesis much more rapidly than the normal callus does. And so that's involved somehow, but no, we haven't. It's entirely, all that callus stuff and regeneration is entirely the work of 3 people, one of whom we finally let graduate, so he'll be moving along before long, and 2 of whom are still in the lab. And one of these days, we're going to have to let him go. Yeah. Sure.
So what comes first in the callus regeneration? Which root marker is turned on first?
In callus regeneration?
Yes, or in the regeneration.
I don't remember.
So WOX5 or one of the...
I just don't
one
remember.
Of the QC markers?
I could look it up if you want to take a minute, because we've got a manuscript for it that's on its way to publication, but I don't remember.
To what degree are epigenetic factors? Another dumb question, I
Oh,
guess.
no, it's a good
Epigenetic questions.
That's what Kaoru is working on now, because most likely there are epigenetic states that get established early that play forward into this that we ought to learn something about. And she's taken all the various candidate genes from curly leaf on and is looking to see what sort of regeneration you get, callus formation and regeneration. And some of them really do have some striking effects. So she's doing it. Yeah.
I was wondering if your modeling is at all dependent on initial cell number.
Cell number.
I mean, do you have a cell number component in your modeling?
Yeah, when we do the meristems, we actually try and use a realistic number of cells. And you can get different results. Maybe I'll get a chance to talk about it tomorrow when I talk about phylotactic patterning. But the number of cells you have in different domains changes the phylotactic pattern. But other than that, we've just used pretty much artificial substrates for most of this modeling. Even the meristem models are really 2-dimensional. We have not gotten to 3-dimensional modeling yet. And we're even struggling with 3-dimensional image processing. There's some frontiers in this stuff that we have not crossed yet. So our models are very simplified, but they do requires some sort of cellular substrate on which to work because the cells are signaling each other. So they're never continuum models.
Since you've been looking at individual meristems quite in some detail, I was wondering how much wiggle, non-mathematical, because once you have an equation, I didn't see any changing parameters in your equation. Not that I'm that good at mathematics.
We have changing.
But so, how individual is each single meristem?
I don't know.
Once you hammer them, you know what I mean?
Oh, they're all
Like,
the
for instance,
same.
if we look at this guy here, that you have self-perpetuating himself. Is this pattern of cell division always the same? In this model it should be. Or do you have a variation?
It's so hard to establish the exact pattern of cell division that we don't really know, but we haven't seen anything that would tell us that it's not very generally the same. Now, if you were to put them in brighter light or in a colder temperature or any of those sorts of things, it could all change. And if we could get a little bit more automated with our image processing, I mean, we did a complete clonal analysis of the floral primordia and the L1 of the meristem, the epidermal layer, that Venu did when he was a postdoc at my lab. It took him about 2.5 weeks of pasting little colored dots onto the movies of the meristem and then going back and counting all the dots. And you can't persuade somebody to do that twice. There's no reason why a computer couldn't do that, and we actually have an image processing team who's working very hard on this, but there's this series of issues that are not really solved in computer vision that we have to get over for this. We sent a grant into NIH to try and get some of this done. The review said, "This is too innovative to be funded by this program." That's a verbatim quote. And so as you can see, it's not so easy to get this stuff worked out. We're trying. And anybody wants to join us, we need everybody. We can't do this alone. Or even with our vast array of collaborators, which is vast, and who do most of the work. Yeah?
You show Wuxia as, in a certain Wuxia as positive variability parameter. Is that a direct thing or is it just an expansion?
It's just an expansion. It's not even an expansion. It induces peripheral zone cells to de-differentiate. That's what these experiments showed. There's a lot of things in the literature of this subject that, people do RT-PCR experiments. You turn up Wuxia, you get more clavata-3. People seem to think that means you're activating the clavata-3 promoter, but that's really not what's happening at all in this case, and probably not in any of these cases that are unexplored either. It's some combination of how many cells are making this stuff and how much each cell is doing it. And you need this sort of live imaging to sort that out, and it, by and large, hasn't been done, which is why I promote this live imaging sort of approach, because it tells you what's happening, which otherwise you don't know, even though you can still publish it, apparently. Yeah. Anybody else?
I think maybe we can have other questions in the discussion before dinner.